There we go. Hi, everybody. Today I'll be going over the pre-lab talk for the Bonding and Metal Chelation lab. So normally this lab was about molecular geometry and VSEPR theory, valence shell electron pair repulsion, talking about the way molecules are shaped. And if you're in the lecture, you'll probably get into that topic. But as far as experiments for that, basically all we could do was have you guys fill out a whole bunch of worksheets and maybe make some models, which you definitely can't do from home right now. So we actually came up with a different one getting into the concept of titration, which we'll use in the next experiment to do a little more complicated version. So I wanted to go through kind of bonding and teach you that technique. So types of bonds. There are really two types of bonds, ionic and covalent, and some of those have some subsets. Ionic bonds involve the transfer of electrons from one atom to another. These usually occur between metals and nonmetals. So for example, sodium chloride, the sodium loses an electron, becomes +1, the chlorine gains that electron, becomes -1, and then that electrostatic force holds them together. These don't have to be one-to-one transfers. You can have something like an iron 2 and an oxygen that gives up 2 and gains 2. You can have things that are off, for example, like aluminum chloride, where aluminum will give up 3. Each chlorine gains 1. You'll need 3 chlorines to balance everything out. So those are all the different kinds of options you can get with an ionic bond. Covalent bonds are involved when you have the sharing of electrons between two atoms. So both atoms retain control of those electrons. They're shared between them, whereas ionic gives up or takes from another atom. And there's a lot of subsets of covalent bonds. One of the major ones is a polar covalent bond. So while these electrons are shared between the two atoms, each nucleus might pull on those electrons with a different strength. And so if you think about a huge football linebacker playing tug of war with, a little 8-year-old, the big linebacker is going to be pulling harder and probably going to pull that little kid across the line. And that's what a polar covalent bond simulates, is one of those nuclei pulls stronger and moves those electrons closer to it, but not enough to take them from the other atom, so they're still shared. I'll show you guys how to look at that in just a minute. There is something that comes up called hydrogen bonding, which actually is not an official type of bond. Bonds are what hold molecules and compounds together. Hydrogen bonding occurs between molecules to kind of hold the bulk together, but is not an actual chemical bond. It's what's called an intermolecular force between two molecules. Hydrogen bonding will occur if you have a hydrogen bound to nitrogen, oxygen, or fluorine, and then the hydrogen bond force interacts between those things, and I'll show you in a minute. So an ionic bond, like I mentioned, you can have calcium, which will give up these two green rings. Those are 2 electrons. It gives those up to the oxygen, which is why they're solid over here. And what that leads to is something called the octet rule. Every atom to be stable wants 8 electrons in its outermost shell. So if calcium gives up these 2, the next ring down has 2, 4, 6, 8, which makes it stable. When these 2 are given up to oxygen, oxygen has 2, 4, 6, 8 as well, making it stable, which is why this bond occurs. And then calcium would become a 2+, oxygen would become a 2-, and that positive-negative attraction is what pulls them together. Covalent bonds, like I mentioned, are where you're shared. So for example, the fluorines will each share 1 electron. And if you look around the blue fluorine, you have 2, 4, 6, that green and blue electron are shared. So they count for both atomic centers. So the blue fluorine has 8. And if we do the same thing for the green fluorine, the 2 shared, 4, 6, 8. So both of these atoms end up with 8. The difference is these electrons are shared between them instead of being transferred like an ionic bond. As I mentioned, you can look at the polarity of a covalent bond. If you have something that is the same atom on either side of that bond, obviously they'll pull with the same strength, and you'll end up with an equal electron distribution. So if you see this kind of gray cloud around, that shows that the electrons are distributed evenly. Whereas if you look around the HCl bond, those electrons are primarily centered around the chlorine side, and there's much less on the hydrogen side, which is why that side of the cloud is smaller. And if you see these little symbols, that's the Greek letter delta. And so in this case, it means partial. So we have a partial positive side and a partial negative side. And those partial positive and negative will actually attract other polar covalent molecules and help hold the whole bulk system together. Like I mentioned, there are hydrogen bonds. Again, they are not a true chemical bond. The hydrogen bond is the interaction between the hydrogen of this water molecule and the oxygen of a separate water molecule. You only get hydrogen bonding if hydrogen is covalently bound to nitrogen, oxygen, or fluorine. So if we look at water, we do have an OH bond covalently, which makes a polar covalent bond where the hydrogen side is slightly positive, the oxygen side is slightly negative. So like a polar covalent bond, the slightly positive hydrogen is attracted to the slightly negative oxygen of something else, of a different molecule. And that attraction helps hold all of the water molecules together. But again, because this occurs between separate molecules, that is not a true chemical bond. It's what's called an intermolecular force. So the last kind of bond that you don't normally hear too much about in introductory chemistry, but it's actually a really interesting bond and has some implications for the human body that I thought, as primarily nursing majors, you guys would find interesting. A chelation bond is also called a coordinate covalent bond or a coordination bond. They are the same, just like a covalent bond. They have 2 atomic centers and 2 electrons are shared. The difference is with the fluorine-fluorine bond we had earlier, 1 electron came from each fluorine to come together and be shared. In a chelation bond, both of those electrons come from 1 atom. So if we look at this nitrogen-boron bond we're forming, nitrogen has these 2 red electrons that it's going to share with boron. So if we look at this coordinate bond, those 2 shared electrons are both red because they both came from the nitrogen. Boron, in this case, did not share any of its electrons. Whereas if we look at the nitrogen-hydrogen bond, we have a red and a green, meaning 1 electron came from each. Or if we look at the boron-fluorine bond, we have a pink and a blue, meaning 1 electron came from each. So those are normal covalent bonds. The fact that this nitrogen-boron bond is 2 red electrons because they both came from nitrogen is what makes this a chelation bond. And chelation bonds occur between usually a metal center, or at least a very small electropositive atom, and another molecular compound called a ligand. A ligand is just something that attaches through chelation bonds. And the number of times it attaches tells you what type of ligand it is. For example, monodentate, bidentate, tridentate, tetradentate, pentadentate, hexadentate are all different types of ligands. And all that changes is that prefix, and it's the same kind of prefixes you've been learning. Mono is 1, bi is 2, hexa is 6, and so on and so forth. So if we look, the nitrogen here that's part of ammonia coordinates to this cobalt metal center 1 single bond, which makes it a monodentate ligand. So it shares of electrons, 2 electrons total, and attaches to the metal center at 1 point. We jump over here to the other side. We have, again, an ammonia center, but this whole molecule is linked together. So this molecule attaches to a metal at 2 points, making it a bidentate ligand. Again, donates 2 pairs of electrons, 4 electrons total, attaches to your metal center at 2 different locations. And that's all ligands do. So hexadentate, to scale all the way up, is one big giant molecule that wraps all the way up around a metal center and attaches at 6 different locations. That is the most locations a ligand can attach to. It leads to tie this back to molecular geometry, it leads to what's called an octahedral geometry. And so we have this big, large molecule attaching at 6 points, meaning it donates 6 pairs of electrons, or 12 electrons total. And the hexadentate ligand is actually what we're going to be looking at today. The specific ligand we're looking at is called EDTA, or ethylene diamine tetraacetic acid. So if you know organic chemistry, which again, I know most of you haven't gotten to, but you can actually break this name down to figure out exactly what EDTA looks like. The important thing about EDTA is if it is fully protonated, meaning all of these hydrogens are on these oxygens, it is a bidentate ligand. 2 nitrogens in the middle can still attach to a metal center. But if you deprotonate it and remove these hydrogens, again, if you look at EDTA versus EDTA4-, the difference is this OH group is an OH group here. On EDTA4-, it's an O- group. And so it's the same thing on all 4 sides for the tetraacetic acid part. When you fully deprotonate it, all of these O- groups can also attach to a metal center as well as the nitrogens. So for example, you can get EDTA chelating around an iron center where the 2 nitrogens attach and all of the O- groups also attach. And so EDTA is a very good chelator, which will go in and find metal ions in your solution and wrap them up and basically pull them out of solution so that they can't interact with anything else. And that's the titration we're doing. But I said chelation chemistry matters in the human body. A lot of you guys are also nurses, so why do you care about chelation chemistry? Well, hemoglobin works the exact same way. If you look at the hemoglobin subunit over here on the left, it pulls an iron 2 ion into the center and chelates through these 4 nitrogen locations. So again, just like what we're looking at, we're going to be wrapping iron 2 up with EDTA. Hemoglobin does the same thing. That's why iron in your bloodstream matters, is that iron goes into your hemoglobin to help it bind. Now, a lot of you probably know that hemoglobin carries oxygen through your system, and there's no oxygen involved here. Well, if you look at the actual protein structure where this hemoglobin unit comes from, it's part of this huge big chain, and you actually get another amino acid residue that comes along the bottom to chelate to site 5, basically. When this happens, it only leaves one spot above the iron open and actually makes it very attractive for oxygen to bind to. So oxygen from your lungs that you breathe in that gets into your bloodstream will see this very attractive iron and go bind to it. However, that bond is specific to certain human body characteristics of that location. So when this hemoglobin unit moves somewhere else in your body, say into your muscles, where if you're working out, there's less oxygen, those conditions change and the oxygen pops back off, which is why we have this double-sided arrow in the middle here. So what happens is the oxygen sticks to that iron, the whole hemoglobin unit moves elsewhere in your body, the conditions change, and that oxygen pops off. Now that oxygen is in a different part of your body that your muscles and other cells can use to perform respiration and other bodily functions. But all of it is dependent on this iron ion being in the center and being chelated by this hemoglobin unit. So again, chelation chemistry does actually affect the human body, which I found really interesting when I first learned about it. So what we'll be doing today to kind of simulate this chelation chemistry and to teach you the titration technique is we'll be doing a titration, or I guess you'll be watching me do a titration. So all a titration is the slow addition of one solution of known concentration to a known volume of another solution of unknown concentration. And you'll be doing this through a Buret. So if you look at this picture, the main parts of a titration setup, you have the Buret with your known concentration, and because you're pouring it out of the Buret, think way back to glassware, you know the volume coming out of it. So you know the volume and the concentration of what's in the Buret. That is your titrant. I'll get into some of that terminology in the next when we really look into titrations. You'll have your titration flask or your Erlenmeyer flask specifically. That's the type of glassware it is. Below it, that you need to know the volume of your solution, but you don't need to know the concentration. That's what the titration will let you determine. And then there's some other supporting objects like a ring stand or a clamp stand, the actual clamp holding the Buret together. Sometimes you'll use a funnel to help add solution to your Buret, a whole bunch of different things. And if you see this little red circle X thing, that's actually called the stopcock. It is part of your Buret, and it's the piece that you will turn, and you'll see me turn in the video, to drain out your solution in your Buret. And so you keep adding what's in your Buret until you get to the end point. And then the question becomes, well, how do you know if you're at the end point? And that is usually measured by a color change. If you're doing an acid-base titration, you can look at pH meters and changes, but we'll be doing, all of ours will be color changes or colorimetric titrations. So the goal of a titration is to determine the moles, concentration, whatever you're looking for of an unknown solution. So again, we're back to measuring unknowns. I did this picture of one of her friends doing a titration one time, especially if you're shorter and you're in a lab and you've got this whole titration set up on the lab bench and it reaches really high up. And again, to measure glassware, you want to be at eye level. Well, if you can't get to eye level, sometimes you can move the Buret down enough to see it. But if you can't, set it on the floor. Sit down, kneel down, do your titration that way so that you can get accurate measurements. That's really what that picture is there for. So again, the glassware. Erlenmeyer flask holds your measured volume of solution of unknown concentration. The Buret holds the solution of known concentration. And because the Buret is very precise glassware, we know exactly how much volume we drain out of it. And so knowing the volume and concentration of what's in the Buret lets us find the moles. As long as we know the chemical reaction, we can use moles of what's in the Buret to find moles of what's in the Erlenmeyer flask. And if we know the volume in the Erlenmeyer flask, we can use those moles and that volume to find the concentration, and that's what we're doing today. So usually, especially for lab experiments, the instructor or the stockroom, whoever's providing the solution, knows the concentration they made. And so in your post-lab questions, you'll be told what that concentration was and figure out how accurate you were doing the experiment. And again, this is me doing it off of some lab data, but you can still find the percent error of how well I did this whole titration by taking the actual concentration you calculate minus the theoretical concentration you're told it was supposed to be. And if you notice there's these lines on either side of that difference, that's just absolute value. So whatever that difference is, make sure it's positive. If it comes out as negative, ignore the negative sign. Divided by your theoretical concentration times 100% to get your percent error. Again, percent error tells you how accurate you were, how close your actual value was to the theoretical value. whereas precision is how close your individual trials are to each other. So remember, all the way back towards the beginning of the semester, we talked about accuracy versus precision. Same concepts come up here again. Percent error is going to measure our accuracy. You can calculate precision through standard deviation. We will not have you guys do that. It is complicated unless you're having Excel or something calculate it for you. But you should be able to look at your trial and say, yeah, those are somewhat close. And again, somewhat being our 20% threshold that we've mentioned throughout the course. So what you'll be doing is, or what you'll watch me do, is filling up your Buret, and we'll do 3 trials. Make sure it gets filled up mostly full. Again, the Buret is read backwards, so 0 is up at the top, 50 is down at the bottom, so fill it up pretty much, and then you'll most of the time be able to do all 3 trials with 1 Buret. If you have to refill it, you refill it, and you'll keep doing the titration until it goes from clear to pink. So if you look at the bottom of this picture, there's a kind of pink solution. What you'll notice me do is it starts clear, and then as you add more, it will eventually turn pink. And so again, I'll talk a little bit more detail about titrations in next week's antacid video. This is really just to get you introduced to it. So this is the data table that will more or less be uploaded, and some of it will be filled out for you. Any of the measured values, as always, I will provide for you, but the calculated values will be up to you guys to figure out. Even something simple like the volume delivered. So what's down here along the bottom is kind of a short key of how to figure out how to calculate each of these pieces. Again, there will be a calculation video that I put up to really go through this for you. These are some of the more complicated calculations, and make sure you're very comfortable with these. The titration calculations we'll do next week for the antacid lab are even more complicated than this. So if you're struggling with these, please reach out to me, let me know. I can get you through these. And I'll go through, again, more detail of this in the calculations video. But that's it for this pre-lab talk. So thank you guys for listening.